today let's talk about extremophiles. So these are organisms that thrive in extreme environments like extreme temperature, salinity, pH, pressure, etc. So archaea are often the most well known to be extremophiles, although not all archaea are extremophiles. But other organisms like bacteria, fungi, eukaryotic microbes like microalgae, etc., they can also be extremophiles. So here, what we're going to do is we're going to go through various environmental factors, like for example, temperature. Then we'll see how different extremophiles survive within a range of different temperatures. So we'll see what adaptations microbe needs to have to survive in extreme high temperatures or extreme low temperatures. Okay, so before we get into the actual extremophiles, what we're going to do is we're going to talk about the different oxygen requirements of microbes, because this is something that I should have covered in an earlier lecture, but we're kind of getting to it now. So again, these aren't extremophiles, but just different ways that we can classify microbes based on their oxygen requirements. So first we have obligate aerobes. Humans would be a great example. These are organisms that require normal, quote-unquote, O2 levels around 20%, which is normally present within our atmosphere. So you can imagine these are organisms that perform aerobic respiration, if you think back to the metabolism lecture. So they're going to require O2 as their terminal electron acceptor in the respiration process so that they can take away the kind of leftover electrons from the electron transport chain. Then you have microaerophiles, which are kind of the same, but they just require slightly lower O2 levels. Next, you have facultative anaerobes. These are ones that they quote-unquote prefer to live in oxygen. So they can produce more ATP in the presence of oxygen because they're doing aerobic respiration, but they can also survive without O2 by either doing fermentation or some type of alternative anaerobic respiration where they don't need oxygen. Then you have your aerotolerant anaerobes. These guys, they don't need O2 at all. So they're fine if it's there or not. It really doesn't matter. So they can live in any kind of condition. And again, they don't need O2, which means that they're using some sort of anaerobic respiration, probably, where they're not using O2 as the terminal electron acceptor, but instead some other thing, like, for example, sulfate or iron 2+. So then we have our obligate anaerobes. These are the ones that they need there not to be any oxygen present because they'll actually die if O2 is present here. So if you recall back to the bacterial culture lecture, we had this thing called thioglycolate broth, right, where we had a liquid culture that we can inoculate with, for example, some patient sample. And then depending on where the microbes grow, right, we can determine what oxygen level that they require. And of course, that can help narrow down to identify what types of bacteria are present within that sample. So for example, if we have obligate aerobes, right, they need oxygen, so they're going to come to the top of the tube where they'll get the most oxygen from the air, whereas obligate anaerobes will go all the way to the bottom because they'll die if they're any closer. So they need to get as far away from the oxygen as possible. Then we have our facultative anaerobes that prefer oxygen, so they'll stay towards the top, but they can still survive without it. So some of them are going to grow at the bottom as well. Then we have our aerotolerant anaerobes, which it doesn't matter if they're with O2 or without O2, which means that they'll just grow uniformly throughout the entire culture most of the time. And then finally, we have our microaerophiles, which require slightly lower levels of oxygen than obligate aerobes. So they're going to go slightly lower down to this level, whatever arbitrary level there is, so that they're not exposed to as much oxygen from the environment. Okay, so let's get into our first environmental factor when talking about extremophiles. So we're talking about osmolarity, which refers to the amount of solute dissolved within a solution. So for example, solute, could be something like salt or sugar that's dissolved in water, where water is the solution, for example. So when we talk about osmolarity, right, we're generally referring to the classic example that we learned in biology, where we have hypertonic, hypotonic, and isotonic environments, where water is, or there's some type of osmotic pressure, right? So water is either trying to get into the cell or out of the cell based on the concentration of, for example, salt within and without, or within the cell and outside of the cell. So let's look at halophiles, right? So halophiles, they thrive in environments that have high salt concentrations, for example, like halophilic archaea that are found in the Dead Sea. So they usually prefer around 2 to 3 molar sodium chloride or just table salt. But this number can vary based on the organism, of course. And of course, this high salt concentration is an example of a hypertonic environment where there's more concentration of salt outside of the cell than inside, which means that the water will be driven out because you're trying to equalize the concentration of salt within the cell and outside of the cell. So that, yep, we said that here because, right, the law of diffusion, going back to that, there needs to be some sort of equilibrium that's reached eventually where there should be basically the system is trying to achieve an equal concentration of salt inside and outside the cell. So the water is leaving because since there's more salt outside, we need more water to go outside to kind of counterbalance the effect of all that salt. That's one way you can think of it. You obviously, these guys, they don't want all the water to get taken out of their they don't want all the water to get taken out of their the inside of their cells. So they have several adaptations, and one of them is called they use compatible solutes. They take these compatible solutes inside of their cells. So what this does is that essentially instead of, right, so if you want to equalize the concentration within the cell and outside of the cell, then one way you could do that is by taking in salt from the environment, right? So that there's less salt in the environment and more salt inside, which will make it kind of even out. But instead of taking salt inside, what they can do is they can use potassium and potassium ions and chloride ions. So this is kind of almost mimics. It's like they're taking in salt without actually taking in NaCl. So they're taking in something that is, I guess, very similar in chemical composition, yet doesn't damage the cells as much. Because if you just take in a bunch of salt, that'll mess with all your proteins inside. So, but of course, if you're taking in all that potassium and chloride, then you need proteins that are somewhat resistant to these things because they can also cause some sort of denaturation in your enzymes. So there are several adaptations that the proteins have to be resistant to these specific ions, or they're also called counter ions. And then other halophiles, instead of taking in solutes, they'll make their own solutes, which will help the proteins, or which means that the proteins don't have to be resistant, right? They can just pretty much function more or less normally. And then these salt-compatible solutes will prevent salt from entering and prevent water from leaving the cell. So here's some examples of compatible solutes that the cell can synthesize on its own. Okay, next let's look at pH. So if you remember, the pH formula is negative log base 10 of the hydrogen ion concentration or the proton concentration. And remember, H plus and proton are the same thing. So essentially, what this means in simpler terms is that if you have a lower pH, it means it's a higher acidity and a greater amount of H plus or protons present. So acidophiles are, of course, microbes or organisms in general that prefer very acidic environments, so pH less than 5. And recall that pH 7 is neutral water, so pH 5, since this is log base 10, right, this is 100 times more acidic than just normal neutral water. So acidophiles, right, since there's so much H plus present within their environment outside of the cell, they need to have very impermeable membranes that prevent the H plus from leaking into the cell. So what they do is they have those impermeable membranes, and then they also have what are called proton pumps, which are proteins that if any H plus gets inside, they can pump it all back out so that they can maintain that relatively neutral environment within the cell. And of course, the reason getting H plus in your cells is bad is because H plus, when it's acidic, right, it messes with the chemical composition of your proteins. And then what that'll do is it'll change how the different amino acids inside the protein react, which means that the overall protein might change in shape and denature, right? And if you denature the protein, then it's not going to do its normal function. So that's why we need to keep all the H plus outside of the cell. And they can also do, instead of taking in H plus, they can take in some K plus, some potassium ion, to generate what's called the Donin potential. So essentially what that does, it makes the membrane more positive, which means that H plus, which is also positive, will kind of get repelled from the membrane and not want to come inside the cell. Some acidophiles can also use molecules that can either neutralize or bind to these H plus, these protons that are coming in. And they can also have acid-resistant proteins and repair mechanisms that can repair proteins that have been damaged by these protons. Okay, next we can go on to alkalophiles. These are organisms that thrive in basic conditions, so low concentration of H plus and high concentration of hydroxide, right, OH minus. So this is in pH greater than 8.5. And these organisms, they often use what are called antiporters, which are these pump proteins within the membrane that take H and they take H plus and pump it inside of the cell so that the cell becomes more neutral in pH. So the pH lowers as these H pluses are put inside of the cell. And in addition, since the outside environment is basic, right? We talked about teichoic acids, which are located in the cell wall of gram-positive bacteria. These acids can help neutralize the base on the outside, and some acidic, some bacteria have S layers, or in some bacteria and archaea have S layers, right? Those protein layers that stick off of the membrane, which are also acidic and can help neutralize the base on the outside. All right, moving on to temperature as our next factor. So we're going to look at high temperature and low temperature. So first, thermophiles thrive in high temperatures that are often greater than 60 degrees Celsius or 140 degrees, sorry, that should be an F here, 140 degrees Fahrenheit. Let's fix that. Great. Okay, and they often, their membranes need to be relatively strong, and they need to be the phospholipids need to be tightly packed together, because the heat is going to try to kind of separate and melt away the bonds between them, right? So what happens is that these thermophiles often have more saturated phospholipids, right? So, and remember, saturated phospholipids are all single bonds, so they have that uniform kind of zigzag conformation, whereas unsaturated, where there's a double bond, sometimes these guys can have these kinks within the chain like this or like this, right? So you can imagine if we have all saturated here, all zigzag, then these phospholipids are going to be able to pack together more easily. So you have these repeating zigzag structures that can pack together more easily. So the phospholipids are closer together, and that helps the membrane become stronger and resist melting. And they also have proteins that are heat tolerant, right? So the heat is going to threaten to break the bonds apart between the amino acids. So these proteins contain many disulfide bonds, which are made by the cysteine amino acid. So disulfide bonds, sulfur-sulfur bonds, which are pretty strong and hold the proteins together, as well as several increased ionic interactions. So you have amino acids that are positive and negative, attracting each other and making that plus-minus bond, that ionic bond. So lots of those within these acid-tolerant, these heat-tolerant proteins. All right, now psychrophiles, they thrive in low temperatures, so often below 15 degrees Celsius or 59 degrees Fahrenheit. And even these microbes have been found in lakes in Antarctica. So you can imagine these are extreme low temperatures, well below 0 degrees Fahrenheit. So here the phospholipids, it's the opposite problem now, right? Now the phospholipids, since it's so cold, they're almost going to kind of freeze together. They're going to pack together and get really rigid. So you want to do the opposite that you did with the thermophiles. So you want to keep the lipids more separated apart so that they don't all fuse together and become rigid. Because you still want things to pass into the membrane so that nutrients can get in and out, right? So here it's the opposite. So the phospholipids are less saturated. That means they have more of these kink double bonds, right? Like this. so that the membrane phospholipids won't pack as well. So they kind of have these, right, these kinks that prevent bonding between these, or kind of prevent interactions between these phospholipids. So the phospholipids are separated more, and they're also shorter, because having shorter phospholipids means there's less opportunity for bonding between the two, let's say, so for example, here and here, right? There can be interactions between these two kind of legs here, but the shorter the phospholipids are, right, we're minimizing the interactions between side-by-side phospholipids and making the membrane overall less bonded together, less fluid, or sorry, more fluid and more flexible. And proteins, right, proteins are also threatening to freeze up in these conditions, so we need them to stay relatively flexible, and we do that by, again, weakening the bonds between the amino acids this time. So now we reduce the number of plus and minus amino acids so that there's less electrostatic attractions. There's less plus minus attractions within the protein. And there are also some proteins or enzymes, antifreeze proteins, which specifically bind to crystals of ice that begin to form, and then they kind of stop those ice crystals from spreading or growing. So there's some intermediate classifications like psychotroph, etc. These are, right, they kind of have the same similar adaptations, but to different degrees. So for example, within the psychophiles, you might have some that thrive from like 0 Celsius to 15 Celsius. Some thrive from like -20 Celsius to 0 Celsius, etc. So that's kind of those intermediate classifications that I listed that are all on the cheat sheet. But the most important ones to know are the thermophile and psychrophile and the specific adaptations here, because even the ones in between, right, are going to have these are the adaptations listed here to some degree. So, all right, next let's move on to pressure, our next environmental factor. So here we'll talk about high pressure environments where these barophiles / piezophiles thrive. So mostly these are located at the ocean floor, where the pressure can exceed 380 times that of the pressure that we feel at sea level. So 380 plus atmospheres of pressure. So since that pressure is compacting these cells down, right, the membrane begins to pack very tightly and become impermeable. And we don't want that because similar to how we mentioned with the freezing, right, we want the membrane to still be somewhat permeable so that things can pass through, nutrients can pass through, our proteins within the membrane can function normally. So these barophiles, they need to offset this tightly packing effect. So again, they contain phospholipids that are shorter and less saturated, just like the psychotrophs here. So that they can resist that membrane packing. Okay, and others, we won't go into the adaptations here because they're not that important, but in case you see these terms on a test, capnophile, these thrive in high CO2 environments. So some of these are organisms that can use CO2 within their metabolism. Then you have xerophiles, which thrive in low water environments. These are mostly fungi. And then you have radiophiles, for example, if you've heard of Deinococcus radiodurans, that's a bacterium that can survive something like, it's like 50,000 times the amount of radiation that a human can survive. So radiophiles thrive in high radiation. Then some extremophiles can check off the box in multiple of these categories. So for example, if a archaean, for example, can survive in high temperature and high acidity, then it can be called a thermoacidophile. So you can kind of mash those words together, and then you can generally call these multiple extremophile organisms as polyextremophiles.